Okay, so clearly there's a lot of depth to what's going on with nematodes. The big initiative there, first of all, was to figure out the quote-unquote connectome. And the connectome is sort of what you see in this picture here. It's just trying to map all the connections between different neurons and then muscles and etc. But usually it's of the nervous system, the connectivity of the nervous system. But this gets complicated because what does it mean for neurons to be connected? It's usually a matter of chemicals transmitting to each other. Sometimes it's electrical, but oftentimes it's chemical. And as we just saw, there's all these different chemicals and receptors, and it's very difficult to get like a clean idea of what this is. And the problem is, we have many different data sets for this. You can see the sort of lineage of various different connectome data sets. And only more recently has there been an attempt to expand this in a few different ways. So one was trying to go just beyond your traditional neurotransmitters, dopamine, serotonin, acetylcholine. Now they're also focusing on what is a huge chunk of the neurotransmission happening, which is neuropeptides. And in some ways, it's an even bigger connectome than the normal neurotransmitter and electrical connectome. There's more connections. Neuropeptides are this whole other class of transmitter chemicals that that still change neural activity, but do so, first of all, over much longer time scales, and also can diffuse into multiple different neurons. So mapping that connectome is a whole different problem. You can see there's even viewers that have been made to view individual cells and their connections. So for example, you pick AIY, you can get all the connections. These are the electrical ones. You can see some chemical ones. And it even tells you where and which data set that came from. Similarly, for the neuropeptidergic connectome, if you do a quick refresh, you can see a very similar thing. And in this case, it's actually telling you which neuropeptides and which receptors are present. And that creates the nature of those connections. So we have all this data. And by the way, if we go back to our simulators, none of this is being really simulated. You can take the connection, but if you don't translate that to the real thing that's going on in that via that connection, the real signaling mechanism, the real biology of what's going on, if you don't have a way of replicating that, then you very well could be missing a lot of the richness of the behavior of nematodes just by not modeling the detail of those individual connections. This also comes into what's called the functional connectome. And so the functional connectome, let's see if there's this example here. But basically, there's already attempts that are trying to basically not just figure out how things are connected, but what it is functionally that each connection does. And so that is looking to answer that question of how do we go from just pure connectivity to like something that actually explains behavior and functionality. Other tools that have sort of been developed for this, one's called NeuroPal. We've made great innovations in sort of experimental tools for live worms recently, and so, or not just live worms, but just getting more detail on in vivo worms. And so one thing that has blown up recently is called NeuroPal, and it allows you via genetic engineering to basically color different neurons based on different expression patterns of different receptors. And so you can actually visually sort of see by the color of their expression which receptors are present and things of that nature. That has been used in a lot of these newer connectome studies, and in fact, this neuropeptidergic connectome definitely used it. you can see that there have been a lot of, more comprehensive studies on what the connectome really tells us about behavior and its connection to also the rest of the worm anatomy. And they've had to do certain graph-like analyses to figure out what is the real functionality of this connectome. Similarly, we have things like the interactome, and even crazier than that, we now have a way to see the neural activity of living, moving worms. And this is due to new studies on how to do calcium imaging, where you can actually track the calcium activity, which is strongly correlated to the neural activity of nematodes. And so you can see these neurons basically lighting up, kind of like where these places glow is us like tracking that activity live on live worms. And there have been more and more improvements to these sorts of things. You can see these have cooler pictures. You can actually see the phosphorescence of calcium activity. And it's only getting better from here. We get higher temporal resolution, higher spatial resolution. And so we're collecting a lot of different data sources. Beyond that, there's a whole group that is more focused on tracking nematode development and how to model that. There's even labs that are tracking genomic inheritance. And so there's a whole lab, Oded Rechavi's lab, which studies how mRNA can get transferred in an epigenetic way to children in the lineage of this worm. And so this worm, I'm telling you, is one of the most studied things in the world. And yet we still, for the life of us, cannot make a decent simulation of the whole thing. So when I was looking at one of my favorite conferences that I'll be attending this year, Artificial Life, I saw that there was a place for art and demos. And I thought to myself, well, given that, I've done some sort of interest projects on these things, which you'll see a lot more of in the following video. But given that I've been so interested in all this stuff and how it comes together, maybe I could, before really making it research, make an interactive demo. And I thought to myself, what really is the compelling part of an interactive demo for this? And to me, it's to demonstrate what would happen if we had actually solved this problem. If we had created a platform, an all-encompassing platform, where you can analyze the data, connect it to the anatomy, all of these things, the simulators, the anatomy, the data sets, if they were all together in one place, what would the scientist of 10 years in the future, let's say, be able to do with an integrative tool that allows you to sort of test any hypotheses, run them immediately in simulations, update your data, change parameters, whatever. And so I'm going to make an interactive demo project called What If We Had Solved the Worm in 2035. So I'll look forward 10 years in the future and try to create an interactive art piece that leverages the real data, as much real data as possible, can run some simulations off of that data, and also, sort of looks into what the future of AI models and things like that can actually give us, the explosion of AI agents that are able to integrate huge data sets and retain all sort of contexts on those data sets while engaging with you in spoken language. This is something completely new that is really may very well facilitate a tool like this and make it accessible for all sorts of researchers. So in the next video, I'll be talking a little bit more about that. I'll be developing out this art piece as I approach the deadline. Now, it doesn't have to be done for then, so for now, it'll just be a lot of ideation, prototyping, and making something that, at least as a pitch, can be really, really compelling.